Emotions have been a source of inspiration for songs, movies, and scientific exploration for years. Everyone has them, whether we like it or not. But where do they come from? Hello, my name is Carlos Diaz, and I'm here today to teach you about one of the most important brain structures involved in emotion processing, the amygdala. I want to introduce the amygdala to you with a story of how I was introduced to the amygdala. I was a high school student in my senior year AP psychology class. We were learning about brain structures and their functions, so my instructor gave us some mnemonic devices to remember how they were connected. For the amygdala, we were told to imagine a Russian MiG, a military aircraft first used in World War II, striking fear into the hearts of those who saw it. So, MiG, fear, amygdala, fear center. The word association was how I came to remember the amygdala as the fear center of the brain. Unfortunately, this connection is somewhat inaccurate, as I will go into later. For now, all I will say is that the amygdala isn't just involved in fear processing, it's also involved in other negative and even positive emotionality, among other things. Much of what we know about the amygdala comes through fear conditioning studies in animals, true, and that's part of the reason for this misunderstanding, but we also know that it works in conjunction with other brain areas to perform a vast array of functions. Anyway, without further ado, let's jump right into the brain and explore the physical characteristics of the amazing amygdala. The amygdala gets its name from the Latin word for almond because of its almond-like shape. Like most brain structures, the amygdala is bilateral, which means that there are two nuclei, one in each brain hemisphere. More specifically, though, the amygdala is subcortically located in the temporal lobe, one of the four main lobes of the brain. To give you a practical sense of where these nuclei are, imagine a line going through both of your eyes and your ears, as if someone fired arrows through your eyes and ears. Gory, I know, but where those lines meet is approximately where the amygdala sits. Diving even further into the brain, neuroanatomists have identified 12 main regions in the amygdala, each with several subregions. Two of the most well understood and important amygdalar subregions are the lateral nucleus and the central nucleus. The lateral nucleus receives sensory information from the neocortex, thalamus, and hippocampus, and projects to various other subregions, including the central nucleus. This central nucleus, then, is involved in our behavioral responses to emotional stimuli, as it projects to several brain regions, including the hypothalamus, the midbrain, the pons, and the medulla, among others. The importance of the central nucleus is further highlighted when we look at the effects of lesioning and stimulating it. Damage to the central nucleus has been shown to reduce, or in extreme cases, abolish altogether a wide range of emotional behaviors and physiological responses. Some studies have even shown that destroying the central nucleus prevents animals from being conditioned to fear something. On the other hand, though, stimulating this region in animals actually produces physiological and behavioral signs of fear and agitation in the animal. Finally, it's worth talking about sex differences in the amygdala because I'm saying these things about location and structure as if they're the exact same in men and women, but in actuality they're not. For example, the amygdala differs in size and rate of maturity in men and women. In fact, the amygdala reaches its full growth potential about 1.5 years earlier in women than it does in male development. Males, however, seem to have larger amygdala, not because we're more emotionally intelligent or anything, because you know that's not the truth, but because the development of the male amygdala takes longer. In addition to changes in developmental timeframes, there are also some hormonal differences in the amygdala of men and women. For instance, the amygdala is full of androgen receptors, receptors that bind to testosterone. Finally, as if sex differences weren't enough, the left and right amygdala within the same person are also different, the left one reaching peak development around 1.5 to 2 years earlier than the right side. Okay, so by now I hope I've made it painfully obvious that the amygdala is a complex little wonder. But the amygdala couldn't do nearly as many things as I say it can do without the help of other brain structures. Which is why, while it's important to study the isolated structure, it's even more valuable to see how it fits into part of a system. Many sensory inputs converge on the amygdala, as it is involved in many of our body's alarm circuits. Kind of confusingly, though, the afferent or incoming fibers are almost always combined with the efferent or outgoing fibers. And you might think to yourself, how is this possible? But it actually makes a lot of sense when you consider the intricate circuitry involved and the feedback mechanisms in the brain. That being said, there are three main ways that sensory information comes to the amygdala. One, directly from the sensory thalamus, which is commonly referred to as the relay hub for sensory information. Two, from the various sensory cortexes themselves. And three, directly from other brain regions, such as the hippocampus and the hypothalamus. Let's first address the first two pathways, which I will colloquially refer to as the fast and the slow pathways. The fast pathway gets its name from the fact that it's, well, fast. Information from an emotional stimulus, and I'm just going to stick with fearful stimuli here since that's where most of the research has been done. But information from an emotional stimulus gets sent to the sensory thalamus and then skips all the higher cognitive processing of the sensory cortex, going straight to the amygdala. For example, if you were walking in the woods and you saw something that looked like a snake, you might feel instantly fearful. That's the information from the thalamus going straight to the amygdala, alerting you to anything that might represent a danger. On the other hand, we have the slow pathway. This pathway follows information from the emotional stimulus to the sensory thalamus and takes an extra stop at the sensory cortexes, which include the visual cortex on the occipital lobes, the auditory cortex on the temporal lobes, the primary olfactory cortex, the gustatory cortex, and the primary somatosensory cortex on the anterior parietal lobes. Sending the information to these regions allows us to further examine the emotional stimulus and then make judgments on it. So, continuing with the snake in the woods example, if what we thought was a snake actually turned out to be a piece of garden hose or a stick or something like that, the sensory information would go through the sensory cortexes first, then to our amygdala. In other words, the cortex makes appropriate adjustments to initial fear responses, suppressing any reactions that turn out to be inappropriate, and we can see from an evolutionary perspective why this would be good for us. Finally, many individual brain structures also project to the amygdala, like the hippocampus, for example. The hippocampus mediates our fear response by giving information about context and is also heavily involved in the storage and retrieval of memories, which might explain why we have an easier time remembering memories with strong emotional context, and inversely, why particular memories can actually trigger strong emotions within us, even years after they happen. The hypothalamus and brainstem are other structures that project directly to the amygdala, as these structures are both involved in, for example, the increased heart rate and breathing that happens when we're around a fearful stimuli, which, facilitates the flow of blood and oxygen just in case we have to run away from a situation. Other notable inputs include the prefrontal cortex, from which we make decisions about what to do in an emotional situation, and the septum, which is involved in reward and reinforcement. Great, so those are the inputs. Now, what about the outputs? Well, again, the amygdala's outgoing and ingoing pathways are pretty interconnected, so we're going to see a lot of overlap here. For example, the amygdala projects to almost every structure I just mentioned. It connects to the hippocampus, the septum, the prefrontal cortex, the brainstem, the hypothalamus, the dorsomedial nucleus of the thalamus, and parts of the entorhinal cortex, too. So, rather than rehash the same structures, I want to emphasize instead two pathways through which information leaves the amygdala, namely the ventral amygdofugal pathway and the stria terminalis. The ventral amygdalofugal pathway leaves the amygdala and continues to many brain regions, including the anterior olfactory nucleus, the piriform cortex, the orbitofrontal cortex, the anterior cingulate cortex, and the ventral striatum, composed of the caudate nucleus, the putamen, and the nucleus accumbens. This pathway is important because it links motivation to responses via the limbic system. It is also a link whereby responses are learned. On the other hand, the stria terminalis connects the amygdala to the hypothalamus, the septal nucleus, and other subcortical structures, and actually forms a loop with the ventral amygdalofugal pathway, as they share certain brain structures in their respective pathways. Also, remember how I said that the most important substructures of the amygdala were the lateral nucleus and the central nucleus? Well, as you can see, the central nucleus is the last stop before information leaves the amygdala and really directs the behavioral, autonomic, and hormonal response depending on where it fires signals to. For example, projections from the central nucleus to the paraqueductal gray matter causes freezing behavior. Projections to the lateral hypothalamus creates sympathetic activation, such as increased heart rate and blood pressure, and projections to the dorsal lateral tegmental nucleus cause cortical activation via the release of the neurotransmitter acetylcholine. Whew, okay, I know that was a lot, but I think the complexity of the amygdala's connections just further proves how the amygdala can't be fully understood if we examine it in a vacuum. Rather, it needs to be studied as a part of a system in order to understand what its critical functions are, which just so happens to be the focus of discussion in Part 3. The most well understood and frequently researched function of the amygdala includes its role in fear processing. Some of the first research into this was done by Heinrich Klüver and Paul Bucy, who, in the 1930s, removed the amygdala of rhesus monkeys and saw drastic effects on behavior, including visual agnosia, an increased tendency to explore items by mouth, dampening of emotional expression, altered sexual behavior, and differences in diet. In humans, this behavior profile, which came to be known as Kluver-Bucy syndrome, also includes amnesia, docility, and a decreased fear response. Additionally, there have been cases of human patients with bilateral amygdala lesions due to a rare genetic condition. Such patients fail to exhibit fear-related behaviors, leading one, patient SM, to be dubbed the woman with no fear. Just listen to Georgetown Associate Professor Abigail Marsh tell one story about SM's fearlessness.
To test whether SM is in fact fearless, the researchers who worked with her took her to a couple of the most frightening places that they could think of in the local area. So one was an exotic pet store, and they offered her exotic snakes to hold in the store, even though she says she's afraid of snakes. They also took her to a place that's, it is at least called the most haunted house in the United States. And they decorate it up for Halloween, and they have people, the staff, dressing up as monsters and things to try to scare people. And the researchers in SM went through with just a regular group. And all the other people who were in the group, as soon as they were in this haunted house that is apparently very spooky, were, huddling together, and they were really slow to go around corners. And SM was taking the lead and saying, "Come on, guys, follow me." And she would be the first around the corners. And when the monsters jumped out and tried to scare her, apparently she would scare them sometimes because she had no fear response to them, which they were not expecting.
Wow. These findings and others like it just reinforce the conclusion that the amygdala plays a pivotal role in triggering a state of fear. In addition to its role in fear conditioning, though, is its role in memory consolidation, modulation, and recollection. In this way, it is similar to the hippocampus, but while the hippocampus is involved in spatial memory, the amygdala is involved in episodic autobiographical memory. In fact, the Markowitz lab has gone as far to assert that the amygdala's main function is to charge cues so that mnemonic events of a specific emotional significance can be successfully searched within the appropriate neural nets and reactivated. These cues allow us to learn from our past via recollection of memories and serves both self-preserving and educational roles. Next up on the long list of functions are attentional and emotional processes. First off, by attention, I simply mean the ability to hone in on certain stimuli while ignoring others. This relates back to our discussion on fear, too, since the amygdala helps us to pay attention to harmful or potentially harmful stimuli and subsequently take action to rid ourselves of that fear, either via flight, fight, or freezing reactions. Historically, this fear example I just used was the only example used, period. However, more research has found that hungry people have increased amygdala activity in response to pictures of food, and people who are very empathetic have an amygdala response to seeing other people. Thus, it's more accurate not to say that the amygdala focuses on fearful stimuli, but that it lets us focus on whatever we seem to care about in that moment. An additional function of the amygdala is its role in social processing. Research by Todorov found that the amygdala plays a role in general evaluation of faces, and research by Kosick and his team found that the trustworthiness in particular is evaluated in part by the amygdala. Furthermore, it appears that amygdala volume correlates positively with both size and the complexity of a person's social network, implying that people with larger amygdala might have more emotional and social intelligence, allowing them to better navigate their social circles and enables cooperation with others. Finally, among the main functions of the amygdala we have is its role in sexual activity. Animal studies have shown that stimulating the amygdala appears to increase both sexual and aggressive behavior, while, contradictingly, one of the symptoms of Kluver-Bucy syndrome, in which the amygdala is lesioned, is hypersexuality. I think that's super interesting and something that more research needs to be done into. But to continue, remember earlier how I talked about sex differences in the amygdala? Well, recent studies have actually suggested a possible correlation between sexual orientation and amygdala brain structure. For example, one study found that homosexual men tend to exhibit more female-like patterns in the amygdala, and homosexual women tend to exhibit more male-like patterns. Additionally, amygdalar connections were more widespread from the left amygdala in homosexual males and heterosexual females, while amygdalar connections were more widespread from the right amygdala in homosexual females and heterosexual males. To finish this section, I want to stop talking about functions of the amygdala and instead switch to dysfunctions or disorders of the brain. The list of disorders in which the amygdala is implicated is unsurprisingly long, given its corresponding long list of roles. In particular, the left amygdala has been linked to social anxiety, OCD, and PTSD. Similarly, patients with both borderline personality disorder and depression show increased amygdalar activity when evaluating emotions in faces, while patients with severe social phobias showed increased activity when shown threatening faces or when confronted with fearful situations. I hope that after hearing about everything that the amygdala is involved in, it makes a little more sense why this brain structure might be involved in all these disorders. Anyway, I could make a half-hour video detailing all of the functions of the amygdala, but I would rather encapsulate the most important ones and then move on to perhaps why there are these misconceptions of the amygdala and other critical analyses of it. This last section of the video is dedicated to critical analysis of the amygdala, so what better way to start than with its most well-known function, fear conditioning. A quick Google search of articles and blog posts about the amygdala wrongly refer to it as the fear center of the brain. Now, this isn't entirely wrong, but as we've seen, the amygdala is responsible for so much more than just processing fear. Sometimes this oversimplification is done for political reasons, but even mainstream news outlets like NBC and CBS are guilty of only viewing the amygdala as involved in fear processing. The amygdala is called the fear center of the brain, in large part because of how it was first studied. A lot of people first came to the amygdala from the study of fear because it's so evolutionarily important, and we know a lot about it in animals. Also, it's pretty easy to test for. Almost every study of fear since has found that the amygdala has been active. However, this fact doesn't mean that the reverse is true, i.e. that every activation of the amygdala is in response to fear. This fallacy is an example of reverse inference, which goes against the idea that just because cognition X, or being afraid, is associated with brain activation Y, or activation of the amygdala, that doesn't mean that activation Y definitely leads to cognition X. Furthermore, a lot of what we know about the amygdala comes from fMRI studies, but fMRIs, in addition to having poor temporal resolution, measure changes in oxygen in the brain, which indicates blood flow, which indicates cortical activity. In other words, it takes a proxy measurement of something which is itself a proxy of the variable being measured, which can make it difficult to get accurate and appropriate data. All in all, though, the amygdala is a fascinating little brain structure. From this little almond-sized collection of nervous tissue comes a lot of our responses to fear, our ability to remember emotional memories, certain sexual behaviors, and a vast array of other functions. Anyway, thank you for watching. I hope you learned something today and gained a greater appreciation of what you now know to be the truly amazing amygdala.